Hello students, today we'll be dealing with the post-fertilization events. After fertilization, we'll see the development of an embryo or embryogeny, formation of endosperm, formation of seeds, fruits. So today, now in this video, I'll be dealing with the formation of embryo, which is called embryogeny. Students, if you look at this figure A and B and compare, you will obviously find some differences. Yes and no. In figure A, this is a mature embryo sac or the female gametophyte. I hope you remember the different parts. Presence of antipodals, presence of the synergids, egg cell, polar nucleus, central cell. But if you look at figure number B, you find only one cell, because after fertilization, the synergids degenerate, the antipodals degenerate. What remains is the egg cell. This is the egg cell, which is haploid in nature. After being fertilized by one of the male gamete, this egg cell transforms into zygote, which is a diploid structure. What are these dotted structures? This dotted area is the endosperm. This dotted area is the endosperm, which provides nourishment to the developing embryo from the zygote. The zygote will give rise to embryo. Embryo needs nourishment. The nourishment is provided by the nutritive endosperm. Now there is one question. Formation of endosperm precedes formation of an embryo. Why? Because the embryo requires food. So much before the embryo is formed, food must be formed. And that is the reason why endosperm formation precedes formation of an embryo. Now look at figure number C. In figure number C, I have shown that how from a single cell zygote we have got one embryo. You must remember, students, as you can see from the diagram also, that the formation of embryo occurs or starts at the micropylar end, because the egg cell is present at the micropylar end. Yes or no? So here we have seen that a much bigger multicellular embryo has been formed at the micropylar end inside the embryo sac. If I draw the diagram outside the embryo sac and try to label it, we find a big cell, which is also called vesicular cell, and a long neck up cell, which is called suspensor cells, or simply suspensor, and a round embryo. This is the embryo. This round embryo is called globular embryo. So this is the embryo. These aren't the embryo. This is the embryo. This is the embryo. Such a shape of embryo is called globular embryo. You can point out the function of suspensor. The suspensor, which consists of 6 to 8 cells, it pushes the embryo into the food-laden endosperm. The embryo requires food, so this suspensor cells, it pushes the embryo right in the center of the endosperm so that nourishment occurs properly. Now this is the globular embryo or globular stage. If we look at figure number D, what can you see? This globular structure has taken the shape of an heart. I've drawn the diagram outside here. The globular structure has taken the shape of an heart, hence this is called heart-shaped embryo. This structure is the embryo, heart-shaped embryo. This remains the same. The suspensor cells and the vesicular cell. Vesicular cell is also called hostorium. If you look at figure number E, figure number E shows a mature embryo. Now you see the development here. The mature embryo, I have drawn it outside here, the structure. This is the mature embryo. Here you can easily find the cotyledons. These two are the cotyledons. Cotyledons. You can find two cotyledons, hence you can easily guess that this embryo is of, yes, obviously it's of a dicot embryo. In case of a dicot embryo, we find two cotyledons. This structure is the plumule that will give rise to the future shoot system of the plant. At the end rises the radicle that will give rise to the root system of the plant. In another video, I will be dealing with the structures of monocot and dicot embryo. But here, in the process of embryogeny, we have seen ultimately how from a single cell zygote we get one globular embryo, then an heart-shaped embryo, since this looks the shape of an heart-shaped embryo, and then a mature embryo consisting of cotyledons. Now, in some cases, as the embryo develops, it uses up the food-laden endosperm. In such case, endosperm is not present within the seeds. Those are called ex-albuminous seeds. And the second one, where even after the complete development of the embryo, endosperm is present, such seeds are called albuminous seeds. Ex-albuminous seeds are also called non-endospermic seeds. Non-endospermic seeds, and albuminous seeds are also called endospermic seeds. We'll be talking about this in another video. So I hope you have understood, students, the process of embryogeny or the development of an embryo in a dicot plant. Thank you.